Hello everyone and welcome. In this video we're going to be solving and explaining all the answers and questions in 9700 Variant 1 Paper 1 May June 2019. So AS Biology, we're going to start. Check out specific questions. Let's start with number 1. A student was told that the actual length of a cell structure is 5 micrometers. The student was asked to state an equation that can be used to calculate the magnification of an electron micrograph of this cell structure. The student used some of the letters Q to U in the equation. Q is the length of the cell structure and cell structure image on the micrograph in centimeters. R is the length of the cell structure image in millimeters, which we use millimeters only. We never use centimeters. Even in paper 3, when we record our answers, you have to, when you carry out the magnification questions, he would ask you, oh, what's the magnification of this image, or what's the actual length? You always have to record the lengths when you are working in millimeters, because then he wouldn't count the working. S equals to 1000 and T equals to 1/5 and U equals to 5. These are just some random values that we can rely on here. So he says which is the correct equation to calculate the magnification. In order to calculate the magnification, again I'm going to draw the triangle. This is a bad triangle, but again it's image length for I, magnification M, and A. I am A. A is the actual length and M is the magnification and I is the image length. So in order to calculate the magnification, I need to get image length divided by actual length. And so far I do have the image length and I have it in millimeters. Now I want the magnification, but I want to, because I know that the actual length is in micrometers, I need up and down to be with the same units. In order to convert from millimeter to micrometer, you have to multiply by 1000. And in case it was the opposite and you want it to be back to millimeters, then you're going to have to divide by 1000. So here I already have the image length, which he said was R. And so R multiplied by 1000, which is, in other words, S. So it's R multiplied by S. And then I can divide that by the image length, or by, the actual length, which is U. So it's R times S over U. Or in other words, because T is 1/5, I can say it's R by S by T, which is D. Moving on. 2. Which features of cilia and root hair cells are correct? Root hairs are correct. So increasing surface area, most definitely root hairs, because cilia are not there to increase surface area, they're there to waft and beat the mucus out of the airways. Cannot be resolved with a light microscope. Actually, the light microscope cannot resolve cilia because, well, they're only surrounded by cells of membrane, and the microtubules that form them are really tiny. They only have 25 nanometer diameter, I believe. And contains vacuoles. What contains vacuoles? Most definitely root hair cells, and more than one present in a cell, cilia. Alright, and basically, because remember that this is, for example, a ciliated epithelial cell that have multiple cilia, but if we look at the root hair cell, only has this one root hair, Okay. What are functions of microtubules? Allow the movement of cilia in the bronchus. As I mentioned, microtubules form cilia, so that's correct. Attachment of the centromeres during metaphase. Attachment of centromeres during metaphase, yes, because microtubules are what forms the spindle fibers during mitosis. And moving of secretory vesicles around the cells, yes, because microtubules form the cytoskeleton of the cell. In other words, the transport pathway. It's like, if you think about it in terms like a subway in real life, here it's going to be like a plan, and it's going to be like there's going to be a whole, I don't know what's the name of this sheet of paper, that I think a blueprint or something, and it's going to have all these pathways and everything. You can imagine these pathways as the microtubules, and they are what guides the vesicles across the cytoplasm and everything. So 1, 2, and 3. And then 4, some cell structures are listed. What is the correct order of these cell structures when listed from largest to smallest? So largest is the nucleus. Yes, the nucleus. And that's because it's 10 micrometers in diameter. And then we have the chloroplasts, because chloroplasts are like 4 to 10 micrometers in width. And then we have the mitochondria, which are 1 to 1.5 micrometers in width. And their length, I think, is between... I think this is the length, and their diameter was 0.5 to 1.5. Remember that 3 is bigger than 1, or chloroplasts are bigger than mitochondria, and 4 is ribosomes. They're either 18 nanometers or 22 nanometers in diameter, which is extremely tiny. So 2, 3, 1, 4 happens to be B. Which cell structures have ribosomal RNA? Ribosomal RNA is found in the ribosomes. And so who has ribosomes out of all these guys? We've got chloroplasts, they have 70S ribosomes. Mitochondrion, it's got 70S ribosomes. Nuclei, the nucleus will contain nucleoli. Nucleoli form the ribosomal RNA because nucleoli, their roles. So if you imagine this is the nucleus, right? And basically it encloses nucleoli, which are basically these dark circular structures that are 3 micrometers in diameter, approximately 3 micrometers in diameter, and they happen to be like 1 or more between each cell, and they're responsible to form ribosomal RNA in order to form the small and large subunits of ribosomes. And so nucleus most definitely contains it. Rough endoplasmic reticulum. The rough endoplasmic reticulum has 80S ribosomes on its surface, and that is why, of course, it has ribosomal RNA. So 1, 2, 3, and 4, A. Now, a cell structure in the macrophage destroys bacteria. Some bacteria stop the cell structure from functioning. Which cell structure in the macrophage is stopped from functioning by bacteria? So what destroys bacteria is actually the lysosome inside it. And so when they stop something from functioning, they most definitely will stop the lysosomes in this case. Because again, when bacteria is going to enter by phagocytosis, let's say, so say that this is the bacteria and it's been engulfed from here, and basically some endocytosis happened, and now we have the bacteria hanging out right here, right? And so basically we've got primary lysosome freshly produced by the Golgi apparatus. They are going to move across the cytoplasm, guided by microtubules of the cytoskeleton, and then they're going to fuse with we call it a phagosome, the vesicle that contains the pathogen, in order to form secondary lysosome, or what we call phagolysosome. And then the phagolysosome is basically where everything is going to be digested. So lysosome is going to be containing hydrogen peroxide to kill the pathogen, and enzymes in order to kill the pathogen. And so basically these enzymes are going to digest the pathogen and everything. So of course bacteria here are going to be like, okay, I want revenge now, right? And so hold on, the erasers. Okay, she's going to want revenge, and so basically she's going to stop the lysosome from functioning. Okay, stops it. Now let's move on. The solution of amylase was added to a suspension of starch. After 30 seconds, 3 samples. 30 seconds, 3 samples of the mixture were tested with iodine solution. So again, amylase was added to starch, and after 30 seconds, we tested with iodine, Benedict's or with Biarritz. Now, which are the expected results? 30 seconds is not enough to break down all the starch. At least, I would not assume that they're going to put such a little quantity. So I would say basically that it's still black, and that this is green because not everything's been converted, and that Biarritz agent is going to be purple because we've got amylase. Amylase is an enzyme. Enzymes are biological catalysts that are actually proteins, and so A is my answer. Now moving on, we have student was carried out a Benedict's test on several different known concentrations of alpha glucose. Which graph shows the results correctly? So if time for the first color change is going to decrease as concentration of alpha glucose is going to increase, because as the concentration of alpha glucose increases, there will be a faster rate of reaction between the reagent and the alpha glucose, and so of course the time for color change is going to decrease. And we know color change is going to be from blue to from blue to green to yellow to red to orange. But if we look at the first signs of color change at the top of the test tube, the diagram shows 3 hexose sugars. This is a repeated question all throughout 2019. Which row shows examples of carbohydrates in which these 3 hexose sugars occur? Okay. So sucrose is going to have 3, which is fructose, and you can identify that by the 5-membered ring structure and the 2 side chains compared to the 6-membered rings over here. So 3. Okay, cellulose is going to contain beta glucose, and who's beta glucose we can identify by looking at the carbon number 1's hydroxyl group. Here it's up, so this is beta glucose, and if you want to memorize it, I like to say it's up, down, and that's for beta glucose. For alpha glucose, I like to say down, down, up, down. That's alpha glucose, meaning it's found in starch. So cellulose is made of beta glucose, starch is made of alpha glucose. Both components of starch are made of alpha glucose, being amylose and amylopectin. And I'm going to give you an extra piece of information right now. Amylopectin forms 80% of starch, amylose forms 20% of starch. Let's move on. So the answer is D. Now, what can occur during condensation of carbohydrates? So a condensation reaction, a disaccharide is produced from monosaccharides, most definitely could happen. Glycosidic bonds are broken. No, that happens during a hydrolysis reaction, which is a breaking down of a covalent bond by the addition of water. And molecules of water are used up. No, molecules of water are going to be released in condensation. And that is wrong. And monosaccharides are produced. No, monosaccharides are the monomers, and they're used to produce the larger things. You cannot possibly produce monomers, right? So it is A, unless you're doing hydrolysis. So what is true about triglycerides? They are hydrophobic. Yes. Are they insoluble in alcohol? Actually, no, they're alcohol soluble. Therefore, the answer is B. So what's the minimum number of carbon atoms in an amino acid? In an amino acid, the minimum number of carbon atoms is 2, because the structure of amino acid is going to be an alpha carbon, the carbon of the carboxyl group. Hold on, this is wrong. Yeah, they prefer to draw it like this, and then an R group on the alpha carbon, hydrogen, nitrogen, hydrogen. So the minimum number is carbon, and is 2 carbons, unless there's more like carbon atoms here, right? And that's like when the maximum number could be reached. In investigation, the same concentration of the enzyme phosphorylase was added to different concentrations of glucose phosphate and incubated at 30 degrees Celsius. Okay. And basically, at 1 minute intervals, 1 drop of the reaction mixture was removed and added to a drop of iodine solution on a white tile. Okay, and the diagram shows the results of the investigation. So here we have the white tile, and basically the drops of the enzyme, I think it's the enzyme known as phosphorylase, was added at 1 minute intervals, and basically was removed and added a drop of iodine. So again, no, actually 1 drop of the reaction mixture was removed and added to a drop of iodine solution on a white tile. Okay, so we take this mixture, like we keep them for a certain period of time, and at 1 minute intervals we'll take a drop and replace them. And so basically, you've got the concentration of glucose phosphate is different, but at different 1 minute intervals you can see basically iodine. Okay. So basically, the color of iodine solution, brown and black. And you can see here that it eventually turns black, showing that the enzyme actually is going to be converting the glucose phosphate into starch, right? And basically, as the concentration increases, the time for it to, turn the glucose phosphate into starch decreases, because you can see here from the first minute, basically like here, from the first minute, this is immediately turned into that, the concentration of 35 milligrams per decimeter cube. Now, what explains the trend in the results of the investigation? So if we were to draw this, the graph should look something like this. Okay, I like to draw when it comes to this type of question. So here, concentration of glucose phosphate, and here is time in minutes. And basically, as concentration increases, the time is going to decrease. But as you can see, we reach a plateau at the concentration of 30 to 35. So we eventually reach a plateau here, right? And so basically, what explains the trend in the results of the investigation? Phosphorylase catalyzes a reaction to convert glucose phosphate into starch. That is true, but the trend is not explained by that. This can all be black, and that would explain the results, but it would not explain the trend. So the maximum rate of reaction is reached at 20 milligrams per decimeter cube, the maximum rate of the reaction. Actually, no, because here it takes 5 minutes, while here it takes only 1 minute to change the color. And the rate of reaction is actually calculated by 1 over time, and the shorter the time, the greater the rate of reaction, right? So the substrate concentration is limiting at concentrations of glucose phosphate at 25 milligrams or less. That is true, because substrate concentration is going to be limiting only when we don't have a plateau. So here you can see the plateau is reached above 35, but below, like at 25, like the first observation before 30 is 25, right? And so below 25, it's always a limiting factor, because as you increase the substrate concentration, the time taken for the color to first change from brown to black is going to be decreasing, meaning it actually has an effect on the rate of reaction. But after 30, it doesn't have any effect. Like you can see, the time remains the same. And at this point here, all the enzyme active sites are fully occupied. And so here, the number of enzyme molecules or the number of free enzyme active sites are the limiting factor, because you need more enzymes to be able to catalyze the reaction at the higher rate for all these substrate molecules. You've got lots of substrate, like in excess. It's no longer limiting, right? And so this is my answer. Let's read the final one. Enzyme concentration is limiting at concentrations of 25 milligrams or less. Actually, the opposite, at above of 25, it will be the limiting factor. Now, 14. What is the definition of Michaelis-Menten constant Km for an enzyme? Km is the substrate concentration that gives half the V max, or half the maximum rate of their action, or half the available enzyme active sites are fully occupied. Okay, that is the Km. And the lower the Km, the higher the affinity of the enzyme for its substrate, meaning the higher the attraction between the enzyme and its substrate, and the more easily the enzyme will combine with the substrate. The higher the Km, the lower the affinity of the enzyme for its substrate, and it's going to be less likely to bind to its substrate so easily, Now, the graph shows how the rate of entry of glucose into a cell changes as the concentration of glucose outside the cell changes. So here we've got concentration of glucose outside the cell, rate of entry of glucose into the cell. So he says, what causes the plateau at X? So here, glucose could enter by two possible ways. Either by facilitated diffusion or by, only by facilitated diffusion, because glucose is large and polar. If it was water, I would have said something else, but this is not water. So we're talking about glucose. And so glucose, all carrier proteins are saturated with glucose. That's the only possible explanation. Again, because glucose can only possibly enter by facilitated diffusion. And when all the available carrier proteins or channel proteins, GLUT2, GLUT1, GLUT4, these are the names of the channel proteins for glucose. When all of them are occupied, then the glucose is going to have like a constant rate of entry. It's just going to enter one at a time, right? So if, for example, you had a line and you were standing in a line, when there's no more space, it's not like basically the rate is going to increase, right? So basically, if you're so let's say this is a line and only one can pass at a time, but here basically let's say you're only one, so of course the rate of entry is going to be way faster compared to when you're three. It's going to be one at a time, so the rate is going to be constant at X, and that's because all the carrier proteins are saturated with glucose. Moving on, the diagram shows movement of substance Z across the cell surface membrane, and here outside the cell, inside the cell, and time. And so here basically you can see that Z is going out of the cell. It's exocytosis. And so you can see over time it's moving towards cytoplasm and not away, and that means it's exo and not endocytosis. How do I know it's not phagocytosis? Phagocytosis and phagocytosis too, they are forms of endocytosis, except basically here we're engulfing pathogens, bacteria, that type of stuff. Phagocytosis, here we're engulfing liquids, but we're not releasing, we're engulfing. That's how you differentiate. Now, Visking tubing is often used as a model during experiments to investigate osmosis in plants. Okay, so what could visking tubing be used to represent? So osmosis is the movement of water from a region of a higher water potential to lower water potential, down a water potential gradient, and through a partially permeable membrane. That is an integral part of the definition, right? And so basically, and when I say there's a visking tubing, the only thing that it can possibly resemble is the partially permeable cell surface membrane. It cannot resemble the difference in constant and water potential, right? So it can only resemble the plasma membrane. So basically, the cell surface membrane, not the cell wall, and the tonoplast. You say, why the cell wall? Because the cell wall is freely or fully permeable to water. It allows water to enter like that, right? But the cell surface membrane, as I mentioned, is made of a phospholipid bilayer, and the phospholipid bilayer has a hydrophobic fatty acid in a core. So it's not fully permeable to water. It doesn't entirely like it. It's just like it's forced to let it in because it's extremely tiny and it's not going to be much of a bother, It's like this guest that you don't want to have, but you know you have to because otherwise it'll be, like shame on you, So cell surface membrane. Tonoplast too is got a partially permeable cell membrane. Tonoplast is the partially permeable cell surface membrane. We call it a cell membrane, not surface, cell membrane of the vacuole. Okay, so that is B. So 18. A scientist stains the chromosome of a diploid plant cell with a fluorescent dye to observe the telomeres. The cell has 38 chromosomes. How many telomeres will the scientist observe in one nucleide during telophase of mitosis? Okay, so during telophase, it's the chromosome is going to only be made of one chromatid, okay? Because throughout mitosis, the number of chromatids that form up chromosomes is going to vary, right? So for example, during prophase, there'll be 2. During metaphase, there'll be 2. During anaphase is when the centromeres split and the chromosomes start to move away from each other like this, right? And during telophase is when each nucleus is going to have only one chromosome with one chromatid, right? Because the nucleus is going to be reforming. And therefore, here, in order to find the number of telomeres are the ends of the chromatids, okay? And so if you multiply every single chromosome by 2, 38 by 2 will give me 76, hence the answer is 76. In prophase, I would have chosen 152 because it's 38 by 4. Now the photomicrograph shows a cell during mitosis. What is happening in the cell? We can see it's anaphase because it looks like what I drew here. Not exactly, but a little bit, right? What is happening in the cell? Centrioles are replicating? No, that happens during G2 in interphase. And if you are not familiar, it looks something. This is a cycle, right? You have this period, and these other period. Oh, I'm sorry about that. Hold on. Okay. So it's like a pie chart. Hold on. So first we have here G1, right? G1 is part of interphase. These are the first phases. So these are like phases in the cell cycle. G1 is part of interphase and it's the first growth period. S phase is where DNA replication happens. G2 is basically the phase where the centrioles are going to replicate, okay? And then we have mitosis. Mitosis is what includes PMAT, or prophase, metaphase, anaphase, and telophase. And anaphase is what we see here, and it's part of mitosis. And then we have cytokinesis, which is basically when the cells split up. Okay, so basically centrioles are replicating. When we establish it's wrong because it's in G2, and spindle microtubules are shortening. The spindle microtubules are shortening and contracting, and that's because there's going to be a hydrolysis reaction happening at the centrosome, which is the centriole bit. Like the centrosome is basically formed of centrioles, right? And so basically this hydrolysis reaction is going to break down the microtubules that are going to be forming the spindle fibers, and therefore the what's called chromatids are going to be pulled towards the opposite poles of the spindle fibers, and that is correct. Okay, so this is right. And then chromatin is condensing. No, chromatin condenses, but that's during telophase. Okay, so that's the last phase of mitosis, so that's wrong. So it's 2 only. Okay, now for this question. Which events are part of mitosis? Interphase, prophase, cytokinesis. Now I've already drew the drawing right here, and as I mentioned, G1, S phase, and G2 are part of interphase. Interphase is not part of mitosis. Mitosis is the mitotic cell cycle. It's got nothing to do with the entire cell cycle. And so mitosis, we're only looking for PMAT. Prophase, metaphase, anaphase, and telophase. And here the only thing available is prophase, therefore it's 2 only. The graph shows the length of the spindle fibers during mitosis. Which region of the graph shows when all the centromeres have detached from the spindle fiber? So the length of the spindle fiber, and basically the centromeres have detached from the spindle fiber. I suppose basically when there's no spindle fiber, then the centromeres will detach from it, right? That's D. So basically 22, short piece of DNA, 19 base pairs long. So 19 base pairs, not 19 bases, because that makes a total difference, was analyzed to find the number of nucleotides in each of the polynucleotide strands. Some of the results are shown. How many nucleotide bases containing C were present in strand 1? Alright, so here we're going to rely on complementary base pairing rule idea. We know that adenine pairs with thymine, and here on the second strand we have 5 thymine, therefore 5 thymine on the second strand will pair with 5 adenine on the first strand, therefore I'm going to write here 5. Right, and then we've got 4 thymine here, therefore we have 4 thymine here. And here we've got 7 cytosine, then we've got 7 guanine. Now we already know there are 19 base pairs, therefore it's like 19 on each strand. So if we subtract 19, so 19 minus 7 plus 4 plus 5 should be 3. Right, so it's 3 cytosine here, and basically, and that's the answer, right? Okay, let's move on. And even if you wanted to do it like on this strand, it's totally possible to find guanine, which is like 3. So to find guanine, which is 3 here, and then according to complementary pairing rule, you'll know that cytosine is also 3. Which nucleic acid bases are purines? Okay, purines are adenine and guanine. Guanine, adenine, C. Pyrimidines are uracil, thymine, cytosine. In a genetic engineering experiment, a piece of double-stranded DNA containing 6,000 nucleotides coding for a specific polypeptide is transcribed and translated. Okay, again, 6,000 nucleotides total. Okay, that makes a difference from 6,000 nucleotide pairs, right? And so what's it called for a specific polypeptide that is transcribed and translated? What's the total number of amino acids in this polypeptide? So first, you just have to say 6,000 divided by 2 because we've got 2 strands of DNA, right? It's a double helix. So 6,000 divided by 2, that's 3,000, right? And 3,000 nucleotides, each 3 nucleotides code for 1 amino acid. That's the code for DNA, right? So basically, each 3 nucleotides code for 1. Therefore, I'm going to divide this by 3, and therefore it's 1,000 amino acids here. So which statements about tRNA are correct? Hydrogen bonds between bases temporarily hold tRNA against mRNA. Hydrogen bonds, that is true. Okay, the base sequence of tRNA molecules are the same as the base sequence of mRNA that is being translated. Actually, that's wrong because tRNA has a complementary anticodon. The anticodon is the sequence of exposed bases at the end of tRNA. And if you want to know the shape of tRNA, it looks something like this, I believe. Right? It has this cloverleaf shape. At one end, it's going to be holding the amino acid. At the other end, it's going to have the code for the amino acid, which is 3 exposed nucleotides, right? And they're going to be very specific, right? And so what's it called? This tRNA is going to move across cytoplasm in order to bind to the ribosome, and in the ribosome, we're going to have mRNA because it's it was basically translation time. And so here, this is going to be complementary to this base, and this base is going to be complementary to this base, and so on. And therefore, it's not the same, they're complementary. Okay, so that's wrong. tRNA translates the base sequence in mRNA in the amino acid sequence in a protein. That is true. It translates the sequence in mRNA into what's called the sequence of polypeptides because it carries an amino acid. Another tRNA is going to come and bind to the next exposed codon, and basically when 2 amino acids are close to each other, they're going to bind together by a peptide bond, right? And so basically here it is the translation part. So that's 1 and 3. Moving on, the photomicrograph shows the transverse section of part of the dicotyledonous leaf. So what are the correct labels for 1, 2, 3, and 4? So 1 is, let's look at that. So yeah, 1 is the xylem, 2 is the phloem, and 3 is the spongy mesophyll, and 4 is the palliative mesophyll. So let's see, we mentioned that 1 is the xylem. Where is the xylem? Xylem and phloem, the spongy then palisade, that's T. Okay, then we've got 27. Some features of present, and transport tissues in plants are listed. We've got what are the features, which of the features are present in the phloem sieve tube elements? Okay, phloem sieve tube elements do not have lignified walls. They do have cytoplasm just on their edges. They could have mitochondria, but mainly the mitochondria are concentrated in the companion cells. The cells are attached to the sieve tube elements because they're going to be carrying out the active processes. Here, only passive processes happen in the phloem sieve tube element. Pits, they do not have pits. Plasmodesmata, yes. And so 2 and 5, is that an option? No, then we're going to have include 3, and that is C. Now, the diagram shows the relationships between phloem sieve tube elements, xylem vessel elements, and companion cells. Which row is correct? 1 is, companion cell, endoplasmic reticulum, phloem sieve tube element. No nucleus, that is totally right. Okay, cytoplasm is not found in the xylem vessel element, and nucleus is not found in the valence. Okay, and vacuole is found here, but is it found in the phloem sieve tube element? That is wrong. Therefore, A is the right answer. 29. Which statement correctly describes the transport pathways in plants? In the alpha-blast pathway, water may move through plasmodesmata. Absolutely not. Water moves in the intercellular spaces and the cell wall in the alpha-blast pathway. But in the symplast pathway, it'll move through plasmodesmata, through the cytoplasm, through the cell surface membrane, but never through the cell wall. And because the cell wall is a dead material, right? Intercellular spaces are not alive, are they? So that's what we call the alpha-blast pathway. It's the dead pathway. The symplast pathway, they move absolutely not. As I mentioned, in the alpha-blast pathway may be blocked by a Casparian strip. Yes, that is true, which is made of suberin. So the photomicrograph shows an external view of the front of a mammalian heart. Which row identifies the position of structures labeled P, Q, R, and S? So P is, let's look at the options, left atrium or right atrium, obviously left. Q, hold on, black. Okay, and Q is the cardiac muscle. Q is the cardiac muscle or the wall of the left ventricle. Okay, R is the Purkinje tissue. I'd like to say that R is a coronary artery because of the way it's branching, and S is the vena cava. Actually, yes, vena cava. You can see it's coming right into the right atrium, so I believe it's B. So what happens during ventricular systole? The atrioventricular node transmits an electrical signal to the apex of the heart. Actually, this is before ventricular systole because this takes some time, so I would not say so. Okay, and the pressure of the ventricles drops below the pressure in the atrium. Actually, no, it increases above the pressure of the atrium, and the atrioventricular valves close and the seminal valves open exactly in order to avoid backflow of the blood back to the atria. That's from the name. Atrioventricular valves, and valves are the valves between the atria and ventricles. We want them to be closed such that when the ventricles contract, blood only goes to the arteries and not back into the atria. Okay, athletes often move from low altitude to high altitude terrain to train for a race. Which statements about the effect of training at high altitude are correct? So here immediately I think about the increase in the number of red blood cells. So let's see. High concentration of carbon dioxide stimulates greater oxygen dissociation. So high altitude we actually have a lower partial pressure of both carbon dioxide and oxygen. Lower concentrations of oxygen stimulate uptake of more oxygen by red blood cells. Lower partial pressure of oxygen stimulates higher production of red blood cells. Exactly. D. Question number 33. The graph shows the oxygen hemoglobin association curves at pH 7.6 and 7.2. Which point on the graph shows the percent saturation of hemoglobin and blood leaving an active muscle? Place of blood leaving active muscle is going to have a low pH because of the high concentration of carbon dioxide released by aerobic respiration. Carbon dioxide released by aerobic respiration is going to undergo, Bohr's effect in the red blood cell and everything, and hydrogen ions are going to be produced, and carbonic acid is going to be formed, and of course carbonic acid is going to reduce the pH, and so obviously the pH is the lower one. Now, what is the partial pressure of oxygen? You can have a lower partial pressure of oxygen and the muscle because they are aerobically respiring, because they are using up this oxygen in aerobic respiration, hence the answer is B. 34. Which row? Okay, 34 is out of the syllabus because it involves smoking. Which statements about bronchioles are correct? They have ciliated cells, they have goblet cells, they have muscle tissue. Actually, they do not have any goblet cells. They could have ciliated cells and they have ciliated muscle tissue. Now, actually, they do have ciliated cells, but in the respiratory bronchioles versus terminal bronchioles will always have ciliated cells, but the respiratory bronchioles are going to have much less ciliated cells. So it's 1 and 3, hopefully. And moving on. Which features are important in diffusion of oxygen out of an alveolus? So blood pressure forces red blood cells through the capillaries. Okay, blood pressure forces red blood cells through capillaries. Actually, okay, that is important, but let's move on to see. Epithelium is permeable to respiratory gases. That is also important for the diffusion of oxygen, and most squamous epithelium are present, also necessary for dissolving of oxygen. Surfactant present to reduce surface tension. Not so important, I would say, but maybe 1, 2, and 3 could help with the diffusion of oxygen, because when you have a high blood pressure, it forces red blood cells through the capillaries, and of course, when the red cells are going to be going through the capillaries, they're going to be squeezed, and more hemoglobin is going to be towards the surface, and so it's easier for oxygen to basically be taken up by the hemoglobin, by oxygen to be taken up by hemoglobin. Now, what's it called? The diagram shows some of the pathogens that cause the disease in humans, and some ways they are transmitted. So you have pathogen and bacterium. Okay, so basically, what is the correct pathogen and method of transmission for measles? Measles is caused by morbillivirus, and method of transmission is by coughing and sneezing. 3 and X, D. Moving on, 38 to 2 rows correct for each disease. Cholera is caused by bacterium called Vibrio cholerae. HIV and AIDS can be transmitted in breast milk and across placenta. That is true. Malaria eradication program unsuccessful. That is true. And this measles is caused by a virus. Totally correct. Okay. Which statements describe myasthenia gravis? Myasthenia gravis is no longer part of the syllabus, but we can also try to answer. Antibodies attack proteins within the body. They do. T lymphocytes are involved in an inflammatory response. The immune system blocks receptors at the neuromuscular junction. They do. And the immune system attacks the central nervous system. The immune system attacks central nervous system. Actually, it doesn't. It's 1 and 3, which is A. Question 40, which row shows the cells that are able to divide continuously and are involved in monoclonal antibody production? So you have to fill out two categories here. They're able to divide continuously and are involved in monoclonal antibody production, because not all cells that are in monoclonal antibody production are able to divide continuously. So examples of cells that we have in monoclonal antibody production, there are cancer cells, which we call B myeloma cells. We have the plasma cells that are produced by the mammal, and we also have the hybridoma cells produced as a result of the fusion of both cells when we put them in a medium together with the chemical known as fusion. So which of them are able to fill out both categories? Let's see. So cancer cells, well, B myeloma cells are involved in monoclonal antibody production and are able to divide continuously. So I'd say yes, cancer cells are filling up both categories. However, mouse B lymphocyte plasma cells, they're not able to fill out this category because yes, they are involved in monoclonal antibody production. However, they cannot divide continuously because they are not cancer cells. They do not have, they're not rich in telomerase, obviously. So we have to make it an X. Hybridoma cells, yes, they are able to divide continuously because we've fused cancer cells, which have a lot of telomerase, with B lymphocytes, which do not have much. And so when we fuse these two sets together, they're going to be able to divide continuously and will be able to produce monoclonal antibodies because the B lymphocytes had the genes to produce these antibodies, which are proteins. And so the answer should be C. So this was it for this variant. I hope this video was useful. If you found it useful, please make sure to share it with other students who might find it helpful as well. Please make sure to leave any notes or opinions that you have about my video and how I can improve this video and what questions you'd like me to re-explain or anything. And any questions that you have in general down in the comments. Also, if you have any resources, like you are done with AS Biology and you'd like to share some resources with the students in order to help them study, that would be perfect. Thank you everyone for watching, and goodbye.